so these are characterization methods. I'm going to talk mainly today about methods by which we characterize the solid phases. I think on Wednesday, Meta's going to talk much more about methods we use to characterize porosity, which is a whole different subject. You can see there's different methods for different things. So the basic idea, I think, as you've already seen, we have a material reacting. At the start, we have unreacted cement, and we have water. And over time, we get the evolution of all these different phases. And depending on the level of complexity we want to have, we can be concerned with all the different cement minerals, all the different CSHs, calcium hydroxide, etc., etc. And what we want to do in terms of characterization is work out how much of all these phases we've got, what their compositions are, what their arrangements are, etc. Right, so in terms of the hydration reaction, we want to characterize phase content and distribution in the matrix. In terms of durability aspects, we can also use this to look at what happens during degradation, how is the microstructure affected by its environment, and what are the mechanisms responsible for degradation. So I've talked already quite a lot, and we haven't said in detail how they work, but I think they're really quite easy techniques to imagine. We have techniques which we can use to follow continuously the reaction, which is mainly calorimetry and chemical shrinkage. What really we're going to talk about here are how we can characterize the solid phases, and we're going to talk about X-ray diffraction. We're going to talk about thermal analysis. I think we're not going to talk much more about NMR than we already have, but we're going to talk a lot about scanning electron microscopy, and I think at the end a little bit about 3D imaging. And before we get into this in detail, I really want to go back to stress this point about errors, because you see, most papers do not contain error bars. We know every single measurement in the world has an associated error with it. I mean, at the very most refined level, we come back to down to the uncertainty principle. But we're not really at that level. We're at the level of much bigger errors, and people tend to ignore that. The problem is that we're often in situations in cements and concretes where we want to have information about very small amounts in a matrix of something else, and that's particularly difficult. So it's particularly difficult to characterize things, for example, at the beginning of hydration or things like that. The second remark is about paste or concrete. So in general, we're always interested in what's going on in the cement paste phase. That's where everything's happening. But if we're looking at concrete, we have 60 or 70% aggregate, which generally dilutes our signal and therefore increases our errors. So that's one good reason why we often use paste rather than aggregate to get better measurements. But we have to, of course, remember that a cement paste is not exactly the same as concrete because of this interfacial transition zone effect, which I showed you earlier. Secondly, if we've got a sample coming from the field, of course, we don't have the option to have that as a cement paste. We just have the concrete, and we have to look at it as it is. And this is the main advantage of the microscopy methods, that you can really focus on the paste regions and put the aggregate regions to one side. And finally, just in this introduction slide, I think what's very important is that usually, if you use just one method, it's not as good as using several methods in combination. And particularly when you know you've got errors, it helps to better identify whether something is a general trend or it's just subject to different errors. So just to summarize the main techniques we're going to talk about, X-ray diffraction is really, I think, the most successful characterization technique for solid materials ever. I mean, it's been around a good 100 years or so. It's widely available. It's quick. It can be quantitative. The one drawback is that really you only get a signal from crystalline materials. And as we've talked about calcium silicate hydrate, we've seen that the major phase we want to often study is this calcium silicate hydrate, which is not well crystalline. Therefore, X-ray diffraction is not the perfect technique in this case. Thermal methods, they're relatively quick and relatively available. They give additional information on water, and they can be applied to both crystalline and non-crystalline phases, but they're not very quantitative, as we're going to see in a minute. Really, they're only used for quantifying calcium hydroxide. NMR has been very successful because it can be used on local materials, because it looks at local order rather than long-range order, but it's very slow. Typically, the kind of measurements I've shown you can take up to a day to acquire the spectra. It's not widely available, and you really need specialist knowledge for interpretation and quantification. And finally, SEM. It really gives you a huge amount of information, often too much information. You can't really process it all or encapsulate it all in quantitative terms. It can be quantitative, but quantification is quite difficult, as we'll come to. It's got this huge advantage of being able to isolate the paste from the aggregate, also to look at microstructures, how they vary in time, for example, microstructural gradients, as you may get from a surface when you have a kind of degradation reaction. It's pretty slow. It's not all that widely available, and I think the biggest problem is you really need to have some experience, and it's often easy to misuse. So let's go back to X-ray diffraction. How many of you have dealt with X-ray diffraction already? Most of you. Most of you? All of you? No? Who hasn't? That's an easier question. Right, two people haven't. Okay, so for those of you who have dealt with it, a bit of revision. The idea of X-ray diffraction is your crystal gives you a regular diffraction grid. And you have X-rays that come in, X-rays that are strongly diffracted in certain directions, and these directions in which you get this constructive interference, you get this diffraction peak, are given by the spacing of the planes according to Bragg's law, which is the most simple interpretation of X-ray diffraction, which imagines that you've got this reflection, and when you have a whole number of wavelengths difference between the X-ray beam here and the X-ray beam here, which is given by this condition lambda equals 2d sine theta, then you get a positive diffraction. What's important to realize is that you need about 10 atomic planes to get a good signal. So when you don't see something by X-ray, it doesn't mean it hasn't got an order. It means that the extent of that order is below 10 atomic planes. And that's basically the problem we have in CSH, that we have an order that is going over about 2 or 3 planes, not over 10 planes. So we don't get a good signal in X-ray diffraction. The second thing, which is the higher level of sophistication in X-ray diffraction, is we have this thing called the structure factor. Now, you don't need to remember this, but what this basically says is that every single atom in the structure contributes to telling how high that diffraction peak is. And this, despite its complexity, this is actually quite a simple equation where we have the indices of the planes HKL here, the positions of the atom X, Y, Z here, and a factor that is related to each atomic species. So this is quite incredible, and this is really the basis of the Rietveld technique I'll come on to talk about later, that you can use this two ways. If you know the crystal structure, you can determine what the peak intensity is. You can also use it to determine a crystal structure if you've got the peak intensities. We usually generally use it the first way because we assume that we know the crystal structures, and then we use it to determine how much of each of those crystal structures are present. So this shows a typical diffractometer. You've got a X-ray generation source here, which fires an X-ray at your sample, and then you've got a detector, which is here, which picks up the signal. This is all automated. Your sample is spinning because you use a powder, but even with a powder, you have a quite low chance of an individual crystal being in a diffracting condition, and you increase that probability of crystals diffracting by having a lot of crystals, as in a powder, and by spinning it round. And then you collect your diffractogram. Unlike most people, including myself, often say, it's not a spectrum, because you're not varying something like a wavelength, you're varying the angle. So it's a diffractogram or diffraction pattern. And you always have a small background, and then imposed on that background, you get peaks corresponding to your different crystalline phases. So if, for example, you had this first crystalline phase here, and then another crystalline phase here, if they have peaks that are well separated, you can distinguish one phase quite easily from another, and you can, by measuring the relative heights of these peaks, determine how much of crystal 1 and crystal 2 you have. So this is a very well-established technique. Now, the problem comes in cements is that instead of having nicely separated peaks like those red peaks and those blue peaks, we've got overlapping peaks. So you see, this is a spectra here, and you see these blue lines are the peaks for Portlandite, these red lines are the peaks for Ettringite, the green ones for A-lite, and so on for B-lite, for hemicarbonate, etc. And when I put them all together, you can see that many peaks have a contribution from several phases. And this is what makes it challenging to determine the amounts of different phases in a typical cement paste. So this is the limits of conventional X-ray diffraction. We have solid solution, which also changes the heights of the peak. So if, for example, if you have a magnesium atom instead of a calcium atom, this changes the scattering intensity of the atoms in those positions, changes the peak's heights. We can have preferential orientation effects, and most importantly, we have these terrible peak overlaps. So this just shows an example of A-lite and B-lite, and you can see there's almost no peaks in either phase that aren't overlapped by the other phase. And for this reason, X-ray diffraction has really advanced considerably with the Rietveld method. Now, the Rietveld method, the idea of it is very simple. You have, on the one hand, you have your experimental pattern, and on the other hand, you make a calculation. And you make this calculation, you know your crystal structures of every phase that's present. You use that crystal structure that you know with the structure factor to calculate the pattern, and then you compare the two. So this is, you know, in quite a simple idea. And in fact, Professor Rietveld came up with this idea in the 1950s. The problem was at the time that there were no computers available, so when you have about 10 phases and in each phase you have maybe 100 atoms, it's just impossible to make the calculation. But now we have computers, so having all this large number of phases, all these large number of atomic positions, we can make these calculations in a matter of seconds, and it's quite easy to iterate these two together to find out how much of each individual phase is present in the mixture. It doesn't finish there, because this is just an iteration, it's just a fit process, and every mathematician knows that the more or every physicist should know that the more parameters you have in an equation, the easier it is to get a fit. Well, here we're talking about calculations where we have literally hundreds and hundreds of parameters. So if we don't well control the way those parameters vary, you can fit almost anything to almost anything. And this is where the skill of the operator comes in, but basically it's a mixture of skill and experience. You start off with simple mixtures. You make sure things working for simple mixtures, then you extend to more and more complicated mixtures, and this is what are called your control files. So the accuracy of your Rietveld analysis depends very, very much on your control files, which are built up over time and experience of analyzing different mixtures. So that's just what I say here. The refinement must be controlled. You can see in this structure factor calculation how many parameters there are. And then, as I say, there may be 100 atoms in a unit cell. You need this crystallographic knowledge to control them. If you get something off the shelf and you don't refine it to the mixtures you're really looking at, you can easily get errors up to 10%. If you do the preliminary work and really refine it, then your errors are considerably reduced, but they don't go to zero. So typically, if you're talking about anhydrous materials, these are the maximum errors. So your error is going to be better than that. So you saw previously when we were looking at the amount of halite that reacted, we were talking about a measurement of about 60% plus or minus 2%. And this goes back to what I showed you earlier, that we can see for different cements the the difference between a correct X-ray diffraction analysis and an estimation, a Bogue estimation, can really be quite large and is not necessarily systematic. So that's for anhydrous cements. We can do the same now with hydrated systems. So in an anhydrous cement, we know all the phases that are there. In a hydrated system, we now have a lot of the phases that don't have an X-ray diffraction signal, notably CSH. So a lot of the phases are there that are amorphous. We can still deal with that if we use a standard, if we always compare our spectrum from our sample with a spectrum from a standard, for example, corundum, then we can back calculate the amount of amorphous material. This is all now quite well-established technology, and this gives an example here of how we can measure the degree of hydration either by X-ray diffraction Rietveld method or by scanning electron microscopy, as I'm going to show you in a short while, and both give very comparable answers, and you can see the intrinsic errors in this system. This is a wider study, so this was just study of one cement over time. This is now the study of 4 different cements at many different times. But again, you can see the same thing, that if you compare the degree of reaction from Rietveld to the degree of reaction for scanning electron microscopy image analysis, you get a very good comparison between the two. The most difficult comparison is at younger ages where you've got low degrees of hydration, and this comes back to the fact that when you're trying to measure small amounts of material in a large amount of matrix, the relative error is more difficult. So just to summarize on the Rietveld techniques, its huge advantage is you can do these measurements extremely quickly. You can get this kind of quantification for complex mixtures in as little as 10 minutes, but if you want a bit better accuracy, it's better to analyze for, say, half an hour or so. The sample preparation is quick and simple, but that doesn't mean that you don't need to take precautions. You can even just saw your surface, but again, there are limits to that. And if you make a powder, you have to be very careful of how you pack it into your sample holder, because it's very easy to get effects of preferential orientation. The big disadvantage is the presence of aggregates. So this shows a diffraction pattern from concrete, and you see this huge peak here. This is quartz, because a lot of aggregates contain quartz. And you see that because you've got this huge peak for the aggregates, all the peaks for the stuff you're interested in all become very small and very difficult to analyze. Other problems, as I said, we can't detect amorphous phases, and we know calcium silicate hydrate is amorphous. Ettringite is a nicely crystalline phase, but when you dry it out, it loses a few molecules of water, and this can cause it to be X-ray amorphous, to not show up well in X-ray diffraction. For that reason, you're best to do X-ray diffraction measurements without any prior sample preparation, without any drying. Our AFM phases are also quite poorly crystalline. We can have phases that tend to be very small crystals that tend to orient in the plane of the sample, and therefore that causes problems for quantification. At the end of the day, as with most things, you need to know what you're doing. And hopefully, when we have the next course in a month's time, we'll be able to spend some time in the lab and really look at these different effects. So, anybody any questions on X-ray diffraction?
Can you modify the sensitivity of the X-ray diffraction so we can get less or more the aggregate?
The only thing you can do is do your scan for longer time. The longer time you acquire your spectrum, the better the resolution. If I go back to a spectral like that, you see, okay, you've got this kind of variation in the background, and therefore it's a question of what's your signal-to-noise ratio. Okay? So if you do your scan slower, so if you acquire your diffractogram over like 10 hours rather than 10 minutes, then you'll get much less background. And then even if you've got quite small peaks, that may be enough to detect it. But it doesn't help much because we've got this, just huge, huge amount of aggregate. What we can do, though, which is another technique we use quite a lot, is if you crush your concrete, then you can basically try to pick out the big bits of aggregate. And then you can be quite effective in getting a material where you enrich the paste phase and you minimize the aggregate phase. Okay? Any other questions? Yeah?
Yeah, in other fields, for example, polymer science, often both X-ray scattering and neutron scattering are discussed together. Are there any people who use neutron scattering for CERN, or does it make so different contrasts which can be seen there no sense in this field, or is it just too expensive?
You're not dealing with, your elements are relatively light, but they're not as light as you've got in polymers. So because you've got elements that have a reasonable mass, like silicon and calcium and everything, you haven't got the same problems you've got in polymers where you've just got carbon and hydrogen. So that's one reason. Secondly, of course, neutron diffraction is not generally available. We want to make these measurements on a routine basis. We don't want to know what's there, we want to know how much. So, typically we want to maybe analyze 100 samples a day. And it's never going to be practical to use neutron scattering on that kind of basis. X-ray diffraction is a real workhorse technique. Okay. So we come on to the next technique, which is thermogravimetric analysis. The idea of this is very, very simple. You just heat your sample at a controlled rate, and you measure its mass. So you start here with your original sample at 100% mass, and as we heat it up, we see a loss of mass. Now, this is a thermogravimetric analysis at just 2 minutes. So basically, we've hardly had any hydration, but we still get a small loss at around 100, which is the small amount of water that's already combined with the sample. And we get another loss here, which is a small amount of calcium hydroxide, probably that was present in the original sample. And then you see as we go 1 hour, sorry, I think that's not, this is calcite. So this would be a cement paste with a small amount of limestone in it, and this weight loss here is the decarbonation of that small amount of limestone. So you see it stays roughly constant through this scan. And as we go on, right, okay, so that was 4 hours. 4 hours, we see a little step there for calcium hydroxide, but the real difference now is at 12 hours. Now we can see this very nicely defined step that corresponds to calcium hydroxide. And we can also see a much more complex weight loss here, which is the hydrate phases. Now, what's important is to see that this weight loss for the hydrate phases starts well below 100 degrees. Okay? In many, many studies going back to Powers, people talk about chemically combined water and physically combined water. And they talk about the chemically bound water as being material that's present at 105 degrees. 105 degrees is huge for these hydrates. You've already lost nearly half the water in your CSH by 105 degrees. So you know, really this is very, very important in reading old papers, particularly where they talk about things like chemically compound water or bound water, because you've already lost a lot of water that's in your hydrates. Now, of course, this also depends on how you've prepared your sample, because you've got to prepare your sample in order to get rid of what's really water in the pores before you start the experiment. And that's also not easy. That's another topic which we'll discuss in more detail when we have the second course, when we look at the practicalities of making samples, is how you prepare your samples for any of these kind of analyses. Nevertheless, if we go on, we see that as we go 24 hours, 28 days, the amount of water combined, you can see your scale is changing each time here, and you can see here between 24 hours and 28 days, we have about 2 or 3 times as much hydration. And as I said at the beginning, we can use this technique to characterize quite accurately how much calcium hydroxide we have in the sample. We can see this nicely defined step here. It's complicated, of course, by the background, because this part here, and even continuing beyond, is the very complex water loss we get from calcium silicate hydrate. So a lot of studies have been made. What's the most accurate measurement method for dealing with this weight loss that's going from calcium silicate hydrate, which is going on in the background? And the best method we can find that compares well with other techniques is this tangent method. And by this we mean that you construct a tangent here which continues this slope here. You construct a tangent here which continues this slope here. You draw a vertical projection at the midpoint of that slope, and you take that step here as a measure of your calcium hydroxide. Okay? And that's just by comparison with model mixtures with X-ray diffraction has been found to be the most accurate way of quantifying calcium hydroxide. So we've got a lot of other information here about our calcium silicate hydrate, etc., which is much better understood by looking at a differential version of this data. So that's thermogravimetric, that's your weight loss here, and then if we differentiate this curve, we get this kind of projection. That was our absorbed water at 2 minutes. This is our calcite peak from the fine limestone. You see, that's designated as ettringite. It might be a bit of ettringite as well. As we go on, you can see we start to form Portlandite here, and then here at about 12 hours, 4 hours to 12 hours is when we get the big formation of calcium silicate hydrate. And this calcium silicate hydrate becomes this rather broad peak here. We go on, and this calcium silicate hydrate peak continues to develop. And you see this complete overlap between where ettringite was originally and where CSH is. So you cannot distinguish ettringite from CSH by this technique. You can see and you can get some qualitative idea of what kind of AFM phases you have from these peaks here, but again, you cannot in any way quantify it because there's much too much overlap between the techniques. However, if you take the weight loss up to, say, 400, that will give you quite a good idea of the total amount of water that is in CSH, ettringite, and AFM phases altogether. Then we have the Portlandite peak here we've already discussed. Here we have the decarbonation. This was a sample with limestone in. It's interesting to note that often you see a double peak here, and often this first bit here is either because your sample has become slightly carbonated during preparation. It can also be slightly carbonated first of all, you can have a slight amount of carbonation during mixing and curing of your sample, but what's more likely is that when you grind it up in a powder and you take it to the DTG machine and you leave it in the DTG machine until it does its measurement, then it can also carbonate. And carbonation is this huge problem in cement chemistry because especially when something's finely ground, it will start to react with CO2 in the atmosphere almost immediately. So, just to summarize that technique, it's very good to quantify calcium hydroxide. It gives you a rough idea about different amorphous phases, but it's difficult to quantify. Incidentally, going back to the effect of temperature, we can see that the effect of temperature actually does very dramatically change the amount of water bound in the CSH we see in this peak here. That's something we're still looking into. It's relatively convenient. Each scan takes a few hours. The disadvantages, and this is particularly a disadvantage here, that every single instrument, every kind of setup changes slightly the positions of the peak. So if you go to a textbook and you look and you say, oh, okay, I expect to see this peak here and this peak here, you can't guarantee that that's going to be the positions you're going to see in your material. And they can vary by a lot, by sort of 50 degrees or so. So, I mean, it's not too much of a problem, because if you're just dealing with cements, everything tends to look more or less the same, and therefore you can work out what things are. But don't slavishly follow temperatures or positions that you read in the textbook. You can see that you have another effect of the amount of this sample. So if we look at this calcium hydroxide peak here, you can see what's the same is the onset of that reaction. But the actual position of the peak depends on the amount. So when we have a very small amount, the peak is here. When we have a larger amount, it occurs much later on. And that's normal, because it's a sort of, you know, you're heating at a defined rate. The phase starts to decompose at a given temperature, but the length of the period of temperature over which it decomposes will depend on the amount. So all of these things are a little bit of precision that you need to be aware about. As I said before, when you're grinding your samples, it's difficult to avoid carbonation. You need to be aware that most of your important hydrates, in fact, lose water below 100 degrees C. So if you pre-dry to 105, you're going to get really complete nonsense. Probably the best way of preparing samples is by exchange with isopropanol. This is a technique we'll talk more about in course 2, which is probably, to our way of thinking, the best way of stopping hydration and yet not too much dehydrating hydrates. Okay, so now we come, anybody any questions on thermogravimetric analysis? Good. Okay, so now we come on to scanning electron microscopy, and this is where I've lost my nice animations. So we have these nice animations which show you how an SEM works. As the animations don't work, I'll have to describe it to you. Basically, you have an electron beam. It scans across your sample in a raster. As the electrons hit the sample, this then leads to a whole series of interactions between the electron beam and the sample, which then emit various signals. Now, I think you can see that, yeah, you can certainly see that in this picture here. So this is a very schematic scheme of electron-matter interactions. You have your electrons coming in here. These are what's called your primary electrons. They go into the sample, and as they go into the sample, they hit the different atoms in the sample, and they undergo a different whole series of scattering processes. The labels here and the various depths are not the depths of generation of those signals, because all those signals, secondary electrons, backscattered electrons, characteristic X-rays, will be generated throughout the interaction volume. But according to the different energy of these signals, this will be the depth from which you can detect those things. So secondary electrons, which I'll explain about more in a minute, are quite low energy compared to the electrons coming in here, and therefore they can only be detected from the very near surface regions of about, that's completely wrong there, because it's way, way less than 0.1 micron. 0.1 micron should be about, for backscattered, it should be about 0.101 micron. Better make that alteration now. Sorry about this. Oops. Okay, so as I said, this is very schematic. We then have the backscattered electrons. Backscattered electrons are very easy to understand. It's just like bouncing a ball. The ball hits the floor, it bounces back. That's the electron. It goes in, it interacts with an atom, and it bounces back. And these, of course, have the same energy, more or less, as the incident beam, and therefore they can emerge from greater depths. Again, I think that's really an upper limit, 0.3 microns, I'd say. Again, it was much less. And finally, we have the X-rays. The X-rays, which are generated by inelastic scattering, as we'll see, which are generated throughout this volume. It's the characteristic X-rays you use to determine the elemental composition, as we've seen earlier, and these are emitted throughout the interaction volume of about 1 micron. So again, we've lost this nice diagram here, but the important points are resumed on the slide. Secondary electrons are electrons from the atom's upper layers, which are ejected and replaced by incident electrons. They have low energy as the interaction is inelastic, and because they have lower energy, they can be collected by a specific detector with a positive charge. So they're not that much influenced by the nature of the electrons. They're very much influenced by the orientation of the surface. So if you have a flat surface, you have quite a little. If you have a very inclined surface, you have a lot. And this is why, hang on, you can get this kind of image where you see the morphology of a product. Okay? So the contrast here is just to do with the orientation of the products. You see, you get this kind of 3D kind of looking images, which is very, very nice for us as human beings to look at. Now, the problem is that this kind of surface you have to get by fracturing your sample. So what you do, you take your bit of cement paste, you hit it with a hammer, you take up a little piece of it, and you put that in the microscope. So that's great, but it's not very representative because you've got a fracture zone. It's not really representative. At early ages, you can see these nice products growing from the surface. But after you get to a few days, as I'll show you later, everything looks the same. So the main uses of this is that, it can give you quite a high resolution. It can show you the morphology of the hydration products. It's quite good at young ages, but it's not representative. You can't do accurate chemical analysis, and you can't do any quantification. But this is a technique, which I've shown you before, where we can follow through how the microstructure develops during hydration. Now, unfortunately, you can pick up any journal with papers on cement and concrete, and you're almost certain to see images like this. I've said it hundreds of times, but I'll say it again. Doing things like this is a complete and utter waste of time. Okay? You're wasting electron microscope time, you're wasting your time, but, it's almost impossible to convince people not to do it. I mean, people say, oh yeah, but you can see all this detail here, I must tell you something. The point is you just can't describe it, you can't quantify it. You can have 4 completely different concretes, and you can spend hours and hours looking at these little details of the products, but you cannot say whether that's representative, it's just a fluke, something happens to be in that area and not in another area. And despite the fact, as I say, I've stood up in these kind of talks and said it hundreds of times, you still get people submitting these kind of pictures, and they've just wasted their time. There are certain exceptions. So, for example, this was a study we did where we had a very particular additive, and this particular additive made a dramatic change in terms of the way ettringite grew. So you can see here we've got very thick crystals of ettringite. Here we've got very thin ones. That was quite useful. But that's really the exception that proves the rule. Most of the time, I really say do not waste your time on secondary electron imaging. It's a waste of time. So now we have, fortunately, an animation that works, and don't ask me why some works and some don't. So this is now backscattering electron. You can see the nice idea here that the electrons come down, they interact with the electrons going around the atoms, and they bounce back. They're sent back in a direction close to that of the emission. They have another different detector. This detector is now around the original gun. And the important thing here is they're highly influenced by the atomic number of the materials they're looking at, and therefore they give you what's called phase contrast. So here's another animation that doesn't work, but what it's supposed to show you is that if you have a carbon atom which has a low atomic number, you get quite a few atoms that bounce back. On the other hand, if you have a calcium atom with a high atomic number, then you get lots of atoms that bounce back. So that's how we get these images. To eliminate any parasite effects of surface topography, of secondary electron imaging, you have to polish your samples. You polish your samples to get something that's as near to flat as possible. And when you have a polished section, then you can really see extremely nicely and in great detail all your different phases. And what's more important, you can then analyze those. So this kind of image we've seen many times before. This is your unhydrated cement. The unhydrated cement, on average, has the highest atomic number. We can see our calcium hydroxide. Here's our calcium hydroxide here. We can see our CSH, etc. And this is all due to the differences in atomic number. So we can write down our phases here. We can calculate by this empirical equation that's been established what should be the backscattering coefficient. This is the backscattering coefficient here. And we can calculate that for the different compounds by the weighted fraction of the backscattering coefficients. And here you can see the numbers, that the brightest phase we expect to see is C4AF, and then CSH is the darkest. We can then use this very reproducible gray level to analyze the images. And it's been very, very long established in 1848 that if you have a statistically representative cross-section, the surface you measure on a surface is exactly equal to the volume in a volume. So people always ask me this question. They say, if you're only measuring the area of that unreacted cement on a smooth surface, how do you know that's equivalent to the volume? Well, I know because this guy Deleuze proved it in 1848. And then later on, some other guy, he proved that if you just took a line and you looked at the proportion of the intercept of those phases on a line, you would also very well estimate the volume fraction. And even if you take points, you will also estimate the volume fraction. So all of these methods are exactly equivalent, measuring the surface on a surface, measuring the line fraction on a line, measuring the number of points that fall within the phase. And, you know, for example, the point counting method, in the electron microscope, we're very lucky. We have this very reproducible contrast, which can be done by a computer. If you do optical microscopy, for example, on clinkers, then you have to have an operator who has to sit there, look at various points, and identify what phase they fall in, because the contrast isn't reproducible. So that's our image. We can then get a histogram from that image. And here we see the histogram of that image there. And what do we see? We see here are the very sharp peaks. This is the anhydrous phases. So this one here is the ferrite. This one here is the alite and everything here. And then we can see calcium hydroxide, which is this peak here. And then we've got this very broad peak, which is calcium CSH merging with porosity. Okay, and unfortunately, I'll come back to this in a minute, we don't see a very sharp cutoff between the CSH and the porosity, and this is because of the nature of the CSH. We've got this very kind of diffuse product with no sharp boundary between a pore and a grain. So for the anhydrous cement, it's really quite simple. We just make a threshold at this point here. We segment the image. We can do a little bit of cleaning up. So, for example, when we segment here, we may decide to fill in the holes, etc. And then we can measure the amount of unreacted cement in this image. And when we measure the unreacted cement in that image, we see in this image it's 16.15%. What's very important is that we have to do then, say, 100 images. Sorry, it's not on that slide. Should be. We should now do 100 images, because in the image next to it, we may have, say, 15%, the image next, 17%, and so on. So it's only by averaging about 200 images we can get a good estimate of how much unreacted cement we've got, and we can use that to calculate the degree of hydration. Calcium hydroxide, you see start to see the problems, that we've now got an overlap of this calcium hydroxide peak with CSH peak. It makes it very difficult to discriminate the calcium hydroxide. We can pull out an image like that, which is quite good for looking at the distribution, but not very good for quantification. You generally find the amount of calcium hydroxide you measure here is much, much less than what you measure by either X-ray diffraction or by thermogravimetric analysis. And finally, we come to the pores. As I said previously, the problem is that we don't see a sharp boundary between pores and not pores. This shows a polished section, a very high magnification. So we're in this kind of image, we're also having a problem with the resolution of the backscattered method. But you can see here, this is a needle of ettringite. This is a needle of ettringite, which would be a few hundredths of a micron in diameter. It's looking rather fuzzy, but you can see this is why we cannot well pick out the pores. Now, and there just shows the secondary electron imaging, same thing. Now, we can't well identify the pores, but what we can do, we can do it in a way that's the same every time. So what we generally do is this. We construct a line here, we construct a line there, and we say, okay, let's take this point here. It's fairly arbitrary, but the question is, does it compare with other methods? And when we do that, we find that, well, it gives you a very good idea about the large pores. You can see the dominance of these hollow shells again. And when we made a series of measurements over several different cement types, and we measured the volume fraction by image analysis, and we compared it by the volume fraction of pores above about 40 nanometers, so we're really looking at the big pores that we could determine by methanol absorption, then we could find that there was a pretty good correlation between the two. So it looks like it should be, you know, it's not the best technique due to this pore boundary effect, but despite that, it does seem to give you reasonable reasonable results which are free of operator error.